You are listening to Professor Megan Workman. Welcome to this lesson about transcription and translation, which are the paired processes that use genetic information stored in your DNA to build your body, one protein at a time. In this lesson, you will learn to describe transcription, and also you will learn to describe translation. At the end of this lesson, you should be able to transcribe and translate a segment of DNA using a tool called a codon chart. So we will go through examples of how you can do that on paper, mimicking what's going on in your cells, and you will learn to define mutations. Here's a cartoon representation of these processes. The top in purple is step 1. This is transcription. It occurs in the nucleus because that's where your DNA is housed. Transcription is when DNA sends instructions for the building of a particular polypeptide. It's saying in these genetic instructions, put amino acids in this specific order. And recall that amino acids are the monomers or building blocks of proteins. Step 2 is called translation. It occurs in the cytoplasm, so it's now outside of the nucleus, and it happens only with the help of a ribosome. The ribosome reads the genetic instructions sent to it from the DNA, and it is the machine that builds polypeptides according to those blueprints. So in the cartoon, you see DNA encoding for an mRNA, which is called a messenger RNA. The mRNA is single-stranded, so it can escape through the pores in the nuclear envelope into the cytoplasm, where it travels to a ribosome, which is like a protein building factory. The ribosome reads the instructions in that sequence of A's, U's, C's, and G's on the mRNA and puts amino acids in a row like beads on a string according to those instructions. So here the polypeptide produced is 3 amino acids long, gly, arg, thr. So let's look at these processes in more detail. All right, first we'll do transcription. So you have your DNA. It encodes for an RNA. The RNA is single-stranded. It escapes through that pore in the nuclear envelope, where it enters the cytoplasm, travels to a ribosome, and the ribosome translates the nucleotide language of nucleic acids into the amino acid language of proteins. So transcription refers to the synthesis or the building of a new RNA molecule under the direction of DNA. Whereas translation is the synthesis of proteins under the direction of RNA. Transcription rewrites the DNA's genetic code into an RNA molecule using the same nucleotide language, where A's pair across from, in this case, a U because it's RNA, and C's pair across from G's. Translation translates this nucleotide language into the amino acid language of proteins. Transcription occurs in the nucleus. Translation occurs in ribosomes in the cytoplasm of the cell. So here is a cartoon DNA molecule. Notice it has 5' and 3' ends arranged anti-parallel to each other. The first part of transcription always begins at a 3' TAC on the DNA. In the cartoon of the DNA molecule, let's find every potential 3' TAC where transcription could initiate. So if I look at the top strand, I put my finger on the 3', and now I'm reading it what feels like backwards from right to left. A, A, T, G, C, T, C, T, A, G, T, A. Okay, so I didn't see any 3' TACs on the top strand. Let's try the bottom strand. So I'm putting my finger on the 3', and now I'm reading it left to right. TAC. Oh, there's one right away. Are there additional ones? TAG, AGC, ATT. Nope, I see one 3' TAC. Here it is. This is the beginning location of transcription on this molecule of DNA, just for example. An enzyme called RNA polymerase that exists in the nucleus attaches here at the 3' TAC. So this is where all transcription begins. At any 3' TAC on the DNA, that could be the start of a gene. The RNA polymerase lays down RNA nucleotides complementary to the template strand of DNA. So when I see a TAC on the DNA, the RNA polymerase is going to lay down a 5'. What goes across to T? An A. What goes across to an A? Well, if it were DNA replication, it would be a T, but since it's RNA, it's a U. What goes across from a C? It's a G, and so on. The base pairing rules apply like this. If you see on the DNA a T, the RNA polymerase lays across from it the complementary nucleotide base. So a T's complement is an A. If we see an A on the DNA, the complementary RNA nucleotide base is a U. C's pair with G's, G's pair with C's. So let's try a problem. Here is an example DNA strand. It's just one strand of the DNA to keep it simple. It's been unzipped and you're looking for a 3' TAC. Here I've bolded that 3' TAC. I don't see any additional ones. So this is where transcription will begin. In other words, I'm going to ignore the A and the T that is upstream from the TAC. Because remember, RNA polymerase, which is the machine in your nucleus that is going to transcribe the DNA into an RNA, complementary new strand of RNA, it attaches at the 3' TAC, so it ignores everything upstream of that. Now, the mRNA strand transcribed is built anti-parallel to the DNA template, so the 3' goes across from a 5'. And notice my A is the beginning nucleotide of my new mRNA strand being synthesized. Ta-da! Hopefully you were able to follow along with that. If not, try additional practice problems that you can find on Google or contact me for some office hours help. Once transcription occurs, that new RNA single-stranded molecule is synthesized. It detaches from the DNA template. The DNA zips back up, goes about its business, and that RNA escapes the nucleus through pores in the nuclear envelope and begins floating around the cytoplasm of the cell. Eventually, it comes in contact with a ribosome, and the next process, called translation, can begin. The ribosome is the protein building factory. It is an organelle found in every cell, both eukaryotes and prokaryotes. So it's not a membrane-bound organelle, but it is an organelle nonetheless. It has a specific function, and this is its function: translation. The ribosome is reading instructions sent from the DNA, and it's fusing amino acids together in a particular order that is encoded by those instructions. It's serving as a catalyst for the creation of peptide bonds, which you can read about more in the assigned reading for this lesson. The peptide bond is a special type of covalent bond that fuses amino acids together in a linear sequence like beads on a string. So what you're seeing in this cartoon image is the complex way that the two ribosomal subunits come together, sandwiching the mRNA in the middle. And you can see the growing amino acid chain going up from the mRNA being built, coming out the top of the ribosome. Translation requires a triplet code, and we call these triplets codons. They're like words contained in the RNA molecule. So the RNA is split into chunks of 3 letters. So here's the first codon, triplet code because there's 3 letters, triplet. Here's the second codon, here's the third, and so on, down the length of the entire RNA molecule. So these codons, these words that are 3 letters long, they don't overlap. And the letters represent nucleotide bases. So it's really 3 nucleotide bases creating these codon words. Since there are 4 nucleotide bases in RNA, A, U, C, and G, you can put them together in every iteration possible, every ordering. It could be AAA, AAG, ACG, and so on. So if you try all combinations, there are 64 combinations. So there are 64 codons possible. That works out because it's enough that each codon codes for a specific amino acid. There are only 20 amino acids. So that means there's extra codons, so multiple codons may code for the same amino acid, but each codon only codes for a specific amino acid. Do you get what I'm saying? Each codon codes for a specific amino acid. So AUG codes for MET, AUC codes for ILE, and so on, until we reach something called a stop codon. Stop codons cause the mRNA molecule to fall off the ribosome, so this ends translation. Even if the mRNA molecule is very long, it continues on, there's more nucleotides downstream of the stop codon, that doesn't matter. Translation ends at the stop codon. This chart is called a codon chart, and this is where I'm showing you every 64 triplet possible. These are all 64 codons, and here you can see what I meant previously by each codon, like U, codes for a specific amino acid, like Phe. But you'll notice that sometimes there's redundancy in this triplet code, redundant meaning that multiple codons might code for the same amino acid. So let's say UUU and UUC both code for the amino acid abbreviated Phe. Highlighted in red are the stop codons. These are the ones that cause the mRNA to detach from the ribosome. So when you're doing translation problem sets, if you come to a stop codon, you're done. You've completed translation. So let's try a problem together. First codon, second codon, third codon, so on. All right, we're going to use this codon chart and the sample RNA strand provided here. They always start with a 5' AUG, and so our first amino acid is always going to be, let's find it on the chart. Oh, it's highlighted in green. That's helpful. AUG, MET for methionine. That's always our first amino acid in every polypeptide sequence. Fun fact. So if you're doing this at home and following along, remember there's a triplet code. So we have to, I like to underline them. You might circle the separate codons. So AUG is my first, CUA is my second, GAC is my third, and so on. And each of these is going to encode for an amino acid. Ta-da! I continued my amino acid sequence until I came to UAA, because UAA is a stop codon. So to highlight that, I just put stop in parentheses, but this isn't an amino acid. This just signifies that translation ends here. All right, so to review what we've learned, transcription occurs in the nucleus. It begins at a 3' TAC. Any nucleotide bases upstream of the 3' TAC are ignored. 3' TAC is the beginning of every mRNA sequence. The second step of the process is called translation. We need to use a codon chart to read which codons are coded by the codons found on the mRNA molecule. So let's try this problem set. It's a little more complex because I've written the DNA 5' to 3'. So in order to find the 3' TAC, I now have to put my finger on the 3', which is over here on the right. And I have to read it, which feels like backwards. T, T, A, T, A, C. There's one. And I'm not going to give you the answers to these. I'm going to let you do these on your own. But there's where you would begin, and you're going to read it in that direction of the arrow, going right to left. Our third problem that you should be able to do is more complex. In this DNA strand, there are multiple 3' TACs, and so you have multiple reading frames. A reading frame is initiated at a 3' TAC on the DNA, and then you keep going until the end of the DNA molecule, and that creates your mRNA strand. But each reading frame can produce its own mRNA strand. Reading frames may overlap one another, so you worry about them one at a time. You find the 3' TAC, and then you find the next 3' TAC and start the process over again. Here's one, here's another. So you're going to be able to transcribe two different mRNA strands of different lengths, and each of those mRNA strands will encode its own amino acid sequence. This is how genes overlap on a DNA molecule. Okay, so now you know how DNA encodes the production of specific polypeptides. But what happens to the polypeptide if the original sequence of A's, T's, C's, and G's on the DNA instructions changed? This is called a mutation. Mutations are any change in the nucleotide sequence, and mutations could be a letter changing to a different letter. Like here, the A changed to a G. Letters may be removed or added to the DNA sequence, potentially during typo during replication. So here a G was added. Now, remembering what you know about reading frames and how there's a triplet code that originates with the DNA because it got transcribed into the mRNA, those codons now are shifted if a letter was removed or added in a mutation. So which of these two mutations is more dangerous, do you think, to the body or to the cell? Well, mutations are more dangerous if they alter more amino acids in the polypeptide that's going to be eventually built from these DNA instructions. So converting an A to a G may or may not cause a change in the resulting polypeptide, but you better believe if you add a G, you've now shifted all of those letters by one space, and that messes up every triplet, every codon that is encoded from this DNA molecule. So when you add or remove a letter, it shifts the reading frame, and that's a much more dangerous type of mutation that could cause the cell to die if that polypeptide was essential for the healthy functioning of the cell. Luckily, most mutations do not occur in genes. So they occur in parts of the DNA that don't encode for polypeptides. That's great because we don't necessarily feel the effects of those mutations because it doesn't affect the polypeptides being built. Mutations occur at a naturally pretty low pace, but they occur randomly. But some chemicals or exposures from the environment can cause mutations to occur at a more rapid pace, and we call these exposures mutagens. These are things like UV light that can penetrate the nucleus of your cells and cause damage to your DNA, cigarette smoke, X-rays, and some viruses can cause DNA mutations. And an example of this is HPV, which is the human papillomavirus, that certain variants of it lead to certain types of cancers. Luckily, mutations are normally not heritable, meaning parents who have accumulated mutations throughout their life, and unless these mutations occur in the cells that ultimately produce egg or sperm, the mutations are not heritable, so you don't have to worry about passing them on to your children. For example, UV light has penetrated my skin cells for my entire existence on Earth, causing the accumulation of mutations in my skin cell DNA, and so I'm at risk for skin cancer. But that does not mean that any mutations I have experienced as a result of that UV exposure are going to be passed on to my children. So I take comfort in that and slather them with SPF. All right, to review, you have learned that transcription occurs in the nucleus of your cells because that's where DNA is. And this is a process that transfers a byte of genetic information from DNA into a single-stranded RNA molecule that can escape the nucleus into the cytoplasm. So the function of the RNA molecule is to take a message from the DNA toward the ribosome, which is the factory that's going to build a polypeptide. That process is called translation. It's performed by a ribosome. It translates the nucleotide language into an amino acid language. So it's following instructions on an mRNA, a messenger RNA, to produce a specific polypeptide by putting amino acids in a specific order and catalyzing those peptide bonds that fuse the amino acids together like beads on a string. You learned how to use a codon chart, so you can now transcribe and translate a segment of DNA in a problem set. For transcription, you know to look for every 3' TAC, and that's the start point of transcription. You know that every sequence of DNA could have multiple 3' TACs, and if you saw that on an exam, you would transcribe every one of them. And then each of the resulting mRNAs from that transcription process, you would use the codon chart to translate the codons, that triplet code, into a sequence of amino acids, translating the mRNA until you hit a stop codon where translation ends. You also learned that mutations are any changes in the nucleotide sequence that may or may not affect the polypeptide encoded by that nucleotide sequence.